Today we are very lucky to have Dr. Linda Van Eldik visiting us from the University of Kentucky. She is a neuroscientist. She is a professor in neuroscience at University of Kentucky. She is, as you can see here, director of the Sanders Brown Center on Aging, co-director of the Neuroscience Institute. And she has dedicated much of her career to developing small molecules that prevent neuroinflammation for a variety of diseases. And she's become a celebrity in the ICH space by bringing this small molecule to trial in humans in a phase 2A trial, the BEACH trial, which is funded by the NIH, the NIA. And you're going to hear about that today and kind of the effort it takes and all the successes that have had to align to get her to this point in the drug development pathway. So thanks so much for visiting us today. We really appreciate it. Looking forward to your talk.
Well, thanks, Chris. Can you all hear me? Usually if I get behind a podium, I'm too vertically challenged. You won't be able to see me. So. All right, so here's my disclosures. I am an inventor on the patents that are going to cover the candidates I'm going to talk about. I'm also a scientific founder of a small spin out from Northwestern where I used to be called Immunochem Therapeutics. I'm on the NIA Council and I have funding. Okay, so just to start, actually before I start, I just want to say thank you guys for inviting me here. And especially on this Remembrance Day, I know a lot of you may have been affected by 9/11, so it's an honor to be here today. So just to get going, if I'm going to be talking about targeting neuroinflammation, it's important to understand what is neuroinflammation. It has a lot of different definitions. Different people think different things, but it's basically activation of the brain's innate immune system. And it's in response to stressors or injury or disease. A lot of different inflammatory responses happen in response to injury and disease. Some of those responses are beneficial. Some of them are detrimental. And so if we want to target dysregulated inflammation, we need to be able to be selective and target the responses that lead to disease progression or injury, bad detrimental effects of injury, rather than the beneficial repair type of responses. And since this is an ICH talk, I thought I'd show this little diagram. Of course, there's multiple mechanisms by which the brain can be damaged after intracerebral hemorrhage. Some of them are shown here. There's a lot of different neuropathological changes that occur that can contribute to damage. The hematoma itself, expansion can cause intracranial hypertension, reduced blood flow, edema, neuronal damage. And I know there's a big effort here for surgical intervention. That's a very important approach. But we have been targeting the pro-inflammatory response to ICH. And we're selectively targeting pro-inflammatory cytokines that get upregulated primarily from activated microglia and astrocytes, and also infiltrating immune cells that can get into the brain because of the blood-brain barrier damage. And we're targeting that inflammation, which can lead to edema and synaptic damage. So this is the process, overall process that our small molecules are targeting. It's essentially dysregulated inflammation. So when the brain is in homeostasis, responses of the glial cells, primarily the microglia and the astrocytes, which is what we study, those are beneficial responses and can keep the brain in homeostasis. But when you have stressors like injury or disease, you can lead to overproduction of those pro-inflammatory cytokines, overactivation, and those inflammatory molecules can be directly damaging to the neurons, to the synapses. And neuronal and synaptic damage is a signal for glia to activate again. And so you can see you could potentially set up this vicious cycle of too much glial activation, too much cytokine production, leads to damage, which then leads to more cytokine production. So we're trying to intervene in that pathway and develop brain penetrant small molecules that can bring that overproduction of cytokines back to the normal physiological levels. We don't want to reduce the basal levels of cytokines because cytokines are beneficial. It's the way your body responds to things and repairs and brings the situation back to normal. So how do we do this? So if you want to develop a small molecule, and I'm only talking about small molecules today, not macromolecular drugs, but even so, it's a very long process. It costs lots of money, lots of time. It's anticipated that 10 to 15 years and over $1 billion just to bring one drug to market. It's super expensive. These are just some examples of some of the grants that we've been funded through over the years to try to develop small molecules. So how did we do it? There's lots of different ways to develop small molecules. This is the platform that we use, and I'll go through this in a little bit of detail just to make sure you understand. This whole project was in collaboration with medicinal chemists and pharmacologists at Northwestern. That's where this project started when I was there. So Martin Watterson and Shakti Roy, very great collaborators. But what we started with was essentially like a Lego piece, a fragment or a chemotype. And then we built it up like Lego bricks. In the very old days, there used to be things, companies would use things like high throughput screening, where you screen hundreds of thousands of molecules. You find the small fragment that has a little bit of activity, and then you build it back up again. We started from the fragment and built up different structures, and we built those up with chemo diversification in mind. We wanted to make drugs. We didn't want to make just the best ligand possible. The chemotype we used is called a pyridazine. It's shown here, the ring with 2 nitrogens in it. And we started with that because many CNS active drugs have a pyridazine as a constituent. And actually, when we started these studies, pyridazines was kind of an understudied type of ring. It's other companies and things were using pyridines and other types of nitrogen containing rings. Pyridazine is nice because you can make changes at any of those 4 constituents there on the ring with the R's. So we would build up a small library of these pyridazine molecules, and then you screen them. And you can do that either screening with a single molecular target. Maybe you want to make an activator or inhibitor of a particular protein you're interested in or a particular receptor you're interested in. Or you do the more classical phenotypic or functional pathway-based approach. In our case, we've done both. So our single molecular target approach was with a P38 kinase, and the phenotypic pathway approach was just looking for compounds that would block selectively pro-inflammatory cytokine overproduction in response to an inflammatory stressor. In any case, you screen, you look for concentration dependent and selective activity of whatever you're screening for. And then we incorporate a pharmacological assessment stage where we don't go right away into in vivo animal models, but we say, okay, does this look like it could turn into a drug? Does this compound, is it stable? Is it chemically and metabolically stable? Does it get into the brain? It's been estimated that 98% of small molecules don't get into the brain. So we had to be in that 2% club if we want to have a brain penetrant molecule against a CNS disease. And then we also did safety screens. Is this compound going to kill your cells or whatever your system you're looking at? Once you get a compound going through there, then you go into the animal models, the more relevant efficacy type of models, look at dose dependence in an animal. At any point, you can go back and have the chemist kind of refine based on your biology that you find, refine compounds, and you can go through and screen again. And finally, ideally, you go into the full drug development where you have the FDA regulated type of preclinical toxicology and eventually into humans if all goes well. So we've actually done 3 compounds that I won't have time to talk about the p38 inhibitor, but that was with the single molecular target approach. And I'm going to talk about the MW189 in particular, which is the one that's in the ICH trial. But the MW151 and 189 were developed with the phenotypical approach. And all three of these drugs have made it into humans, which is really exciting. So they're in either phase 1B or 2A trials. I'll just give you a little bit of summary information of some of the things that we've seen along the way. So these are the two compounds, the 189 and 151, and you can see they differ. They're very close analogs structurally. They just differ in that one position there where there's a methyl in 151 and a whatever that's called, in pyridine, I think, in the 189. I'm not a chemist, as you know. So 151 is actually in an oral formulation and 189 is an intravenous formulation. But this just shows you an example of using an Alzheimer's mouse model where we administered ICV, either beta amyloid, A beta 1 to 42, or vehicle ICV over 4 weeks. And then at 3 weeks, so the injury is already underway. At 3 weeks, we administered compound once a day, or twice, 3 times a week, 2.5 mg per kg orally. Then we stopped the drug and waited another 3 weeks before we did behavior and sacrifice. And we did that because we wanted to make sure that the effect we were seeing was going to be long lasting and not just completely reverse once we took compound away. And you can see in the graphs, this model shows an upregulation of pro-inflammatory molecules, interleukin-1 beta and S100B. And in the black, the compounds are bringing those levels back down toward the vehicle basal level. This model also shows synaptic damage, loss of postsynaptic density 95, which is a postsynaptic protein. You can see in the white with the injury alone, the levels go down, and then with the drugs, it's brought back up. And Y-maze is a behavioral test, a spatial, hippocampal-dependent spatial test. It also protects against the cognitive impairment. So that was great. We've also gone further with these two compounds. We showed in an actual knock-in mouse, an APP-PS1 knock-in mouse, which doesn't overexpress A beta. Again, we block the levels of IL-1 beta, but we don't block the levels of IL-10, which is an anti-inflammatory cytokine. So there's selectivity in terms of what's being blocked. And the middle graph is a traumatic brain injury model, and this looked at dose dependence, and you can see that it's pretty powerful all the way down to 0.5 mg per kg in this animal model. And on the green are just some other preclinical models that we've looked at with 151 or 189. They behave very similarly in different animal models, and you can see all of these models are all of these disease models are injuries or diseases where inflammation has been thought to contribute to progression. The 151 is actually just finishing a trial with people with brain metastases who get radiation therapy. That causes inflammation and they get cognitive impairment later. And it also works in the radiation-induced cognitive decline model was the rationale for why we went into that in humans. So these drugs are kind of disease agnostic. You could potentially use them for a large number of different injuries or diseases. This is an example of a ICH preclinical model. This was done in collaboration with Danny Laskowitz at Duke. And you can see in the rotarod latency when the mice are treated with 189 twice a day for 5 days. There's a significant improvement in the vestibular motor function by rotarod. And I don't show the data, but there was a significant decrease in the cerebral edema that you see in this model as well. So this just summarizes all the preclinical development of 189. It's actually a really interesting compound. It's water soluble. It's extremely stable. I mean, we have a batch of drug substance that was made back in 2014, and it's been tested every year, and it's still perfectly stable. It's like a water-soluble brick, we call it. It's orally bioavailable. It gets right up into the brain, CNS penetrant, very quick. It's not an NSAID. It's not a steroid. It's not immunosuppressive or an antibiotic that may have anti-inflammatory properties. So it selectively targets those signaling pathways that lead to cytokine overproduction. So we don't affect basal levels. It's just the disease-induced upregulation. And it has it has efficacy in a lot of different animal models, as I mentioned. So we also showed in FDA regulated type of toxicology and safety test. It doesn't block any kinase, that at least in these kinome-wide screening parameters, it doesn't block ion channels or transporters or enzymes or these receptors. So it's an unusual mechanism. And it's got great safety in CNS, cardiovascular, respiratory, genotoxicity. And we've finished now the 6-month oral toxicology in rats and 9-month oral tox in dogs. And it's safe at the highest dose that we tested. So our no obvious adverse effect level was the highest dose tested. So our safety profile is much stronger. Our therapeutic window is very high because our efficacy doses are very low and our toxicology doses are very high. So then we decided to go into humans with the 189, and this was done with Danny Laskowitz and Jeff Guptill at Duke, and they did both phase 1 single ascending dose and multiple ascending dose studies. These were in normal adults. It shows on the right-hand side the linear kinetics that you see. And basically, it's very well tolerated in both studies. And based on the PK data in humans, doses of 0.15 to 0.25 would be within the efficacy values in animals. Then there was a really interesting little study. We wanted to see if we could engage our mechanism in humans. And because normal adults have basal levels of cytokines, we couldn't measure cytokines in a normal adult because it wouldn't change. So what they did was a small phase 1A with endotoxin administration to healthy adults, basically LPS. I didn't even know you could do that, but there were only 9 placebo, 9 drug patients, 1 dose of 189, and then a very low dose of LPS, and blood was taken over 12 hours and plasma cytokines were measured. There were 6 cytokines. I didn't think any of them would change with so few subjects, but 2 significantly changed. TNF alpha, which is pro-inflammatory, went down with drug, and IL-10, which is anti-inflammatory, went up with drug. I was really surprised about this, and this is kind of at least initial evidence that it does engage its mechanism in humans. So basically, to summarize where we were in the phase 1 studies, the single ascending dose, the multiple ascending dose, as well as this kind of endotoxin sick patient study, all of these individuals, of course, when you get endotoxin, you kind of get like flu-like symptoms because you get a big inflammatory response. It was safe in all of these individuals, and it brought us to the rationale, okay, let's see if we can go into a first inpatient. And after talking with lots of individuals and, working with different groups, we ended up doing this beach study, which is what we're in right now. This is being done with Dan Hanley. He and I are multiple PIs on an R01 grant that's been funded by NIA, which also in itself was pretty interesting because usually NINDS funds stroke trials. But we convinced NIA that this patient population was relevant because, first of all, they're mostly aging individuals. They're 55 or over. They get a lot of CAA, cerebral amyloid angiopathy, and many of them get cognitive impairment. So NIA agreed, and So we're in the beach study now. This is first in patient. It's an IV formulation. We're targeting patients that have non-traumatic, spontaneous ICH. Their average age is above the age of 65, NIA like that. And substantial numbers, as I said, have both cerebral small vessel disease as well as CAA. They have a lot of comorbidities and medications that we're also keeping track of. So our primary objective is to look at safety and tolerability. This is not an efficacy trial. It's not powered for efficacy. And so through day 7, we're looking at safety as our primary outcome. We have a bunch of secondary endpoints, both PK, hematomal edema, CSF and plasma biomarkers of both inflammation as well as CNS injury, and of course, safety and tolerability throughout. So here is how the trial is set up. A person comes into the ICU or emergency room. They get evaluated and consent and get treated within 24 hours. So they have a CT stability. They have all the standard things. If they meet all the inclusion criteria, they get treated with 189 or placebo. It's random. It's 120 patients total, 60 drug, 60 placebo. They get treated with 189 or placebo every day for 5 days, twice a day, or discharge, whichever comes sooner. Then there's follow-ups. There's a 30-day in-person, 90-day telephone, and 180-day clinic. We have 12 clinical sites. We started out with 10, but we had to increase because it's difficult to recruit. This is kind of a difficult population, but also it's a difficult procedure. There's a lot of PK draws. But so far we've actually recruited 88 patients, so we're getting there. And it's safe. So far it's been safe and well tolerated. So what have we learned so far? So you guys probably all know this because you do these kinds of studies all the time, but I am not an ICH person. And it was interesting to me, the challenges that we faced. So first of all, of course, we're interested in safety. So we needed to make sure that we had real-time, adverse event monitoring, that we had really good education for all of the nurses and the coordinators at the different sites, ensuring that we had data integrity across sites. This is 12 sites was, at least for me, is a complex study. We also had to, since it had to be within 24 hours, we really needed to get people in, screened, identified in terms of eligibility, and get them to consent, or their LAR to consent. So they set up a Viz.AI assisted screening, which automatically would communicate with the PI or the coordinators to say, hey, a patient's coming in. You had 24/7 research team coordination to try to not miss anybody that could be in the trial. And then the pharmacokinetics is difficult. We have several draws after the first dose, and then 12 hours later at the second dose, because it's twice a day. And getting those timings appropriate and ensuring that, they don't take the blood from the same arm that they're infusing the drug, those kinds of things. It was a learning curve. And then also, proactively trying to schedule the follow-ups, making sure that we don't lose people after they are out of the ICU. And finally, trying to adapt to any protocol deviations or operational type of challenges, and managing drug-drug interactions. All of those things have been an interesting learning experience for me, but fun. So it takes a village, of course, and these are just some of the people from my lab and also different collaborators that I mentioned along the way. And of course, our funding agencies, which we couldn't have done any of this without them. So thank you very much. I'm happy to answer any questions.
Thank you so much, Dr. Van Eldik. We're going to switch to a panel discussion for questions. So if you could take a seat here, and then also I'll call up Dr. Fernanda Carvalho Poirez, who is a assistant professor here in neurocritical care, Dr. Michael Waters, who is the director of vascular neurology here, and Dr. Jankowitz, who I don't see. So, oh, there you go. Great. How could I have missed you? Who is director of cerebrovascular at JFK and also co-PI of the MIRROR registry. And so we've been hoping to have a little bit more of a long-form discussion about the topics covered in the talk when we have an in-person speaker from MIRROR Grand Rounds. So feel free to post a question in the chat on Zoom, and we'll also take questions from the audience and have a little bit of a discussion. So, Kenan, do we have any questions in the chat? Okay. Any questions from the audience or panel? Yeah, Fernanda. Yeah.
This was a great talk. Thanks for going through this process of drug development. It's interesting to see like how things go from that side. I was curious to hear about how you ended up getting connected with ICH researchers and how much of the preclinical research in ICH specific animal models and cytokines in the brain or peripherally like went into that, that process of eventually developing the beach trial.
So generally in all of our animal models, I will collaborate with experts who are in the field. So the Alzheimer models that I showed, that was my lab because that's what I do. All of the other animal models were in collaboration with people who have these animal models. And what we would always do is define the time frame of when the cytokines are changing in these different animal models. So like, for example, in our APPPS1 knock-in model, we had taken samples of brain at different times and showed that the cytokines begin to go up about 6 months, and then they rise up to 11 to 12 months. And so when we did our long-term experiment that I showed, we started administering at 6 months as the cytokines were just beginning to go up, and then stopped at 11 months. So we would do that for every animal model. So with the TBI, we knew when the peak, the acute surge and down. And that was how in the ICH trial, we looked at the literature and knew that the cytokine surge after the hemorrhage was between, right away over the first 3 to 5 days. And so that's how we designed a 5-day administration, because that's when the cytokines are changing. And that's when you would want to administer. The neat thing about our drug, the way that it works, is it has a long-lasting pharmacodynamic effect, because if you block the production of the cytokines, which is what we're doing, we block biosynthesis, we block RNA that leads to protein production. If you block that and that cytokine surge doesn't happen, then all those downstream things don't happen, and you don't have to have drug on board anymore. Never got the cytokine surge to happen. And so that's why it's not like a drug that binds a receptor that always has to have the receptor engaged, and you always have to have your drug around. So actually the drug, I think, works much better in acute diseases where you only have a single inflammatory stimulus. In our upcoming Alzheimer's trial, it's a little bit different, because there it's more of a low-level chronically elevated cytokine. So we're going to plan to administer every day for a long time.
It's really interesting that this targets the acute escalation in signaling without interrupting the baseline function of the immune system in the brain, which we basically only have steroids right now. And so with steroids, we're really shutting down almost everything as much as we can, but it creates all these other issues. So it's extremely convenient. What are some other features of the drug that make it particularly convenient for neural administration or maybe even cerebral edema treatment?
Well, with edema, with the stroke treatment, you're in a hospital setting, so it's very easy to administer a compound. One advantage of our compounds for other diseases, like let's say traumatic brain injury, since they're so stable, a soldier could have it in their pocket and they get a TBI or a blast injury, they could just take the drug and, prevent that inflammatory surge. So technically, it's stable for years and years and, still active. So that's one big advantage. The other advantage is it's very selective. So once you stop it, it's not going to be around to prevent the beneficial reparative processes that happen later on. And if it does reduce edema in humans like it does in animal models, that should be beneficial, I would think, too.
Have you looked at, or do you think it's possible to disaggregate the kind of the smoldering chronologic effects of one of these acute injuries versus the acute phase? For example, in BCID, and ischemic strokes. I mean, there must be some initial damage that is maximized, but is there an ongoing process that may be different than the cytokine surge associated with the acute event?
We've started to do some kind of more panels of responses rather than just picking out certain cytokines that are just your standard cytokines and measuring them. And we are seeing some interesting effects on metabolism as well as the cytokines. So it's possible that there may be some beneficial effects that relate to, metabolic effects or mitochondrial oxidation or things like that we really haven't defined. The fact that we don't know the exact way that it works, we know the pharmacological mechanism, but we don't know the exact molecular target, is fine. I mean, FDA approves drugs all the time that don't have molecular targets, but it would be interesting to see what different kinds of signaling pathways are converging and diverging that might have other effects downstream besides just the cytokine overproduction.
Just to elaborate on that, have you seen any side effects? And could there be systemic side effects? I assume this is working on cytokine storm systemically, and it also works in the brain and is also inhibiting inflammation in the brain, but it's not a neural specific molecule, although it does get into the brain readily.
Right. So, I mean, the endotoxin studies show that it can have an effect. It can have effects in peripheral immune responses. And the fact that it gets into the brain quickly, I mean, it doesn't dwell in the periphery very long, but it's not immunosuppressive. We saw that patients can mount a fine immune response against endotoxin. And it's definitely not neural specific, but it may have effects on peripheral immune cells coming in, for example, because it does have some effects on chemokines. So that may be of benefit if you can prevent some of the infiltration of some of the peripheral adaptive immune cells. But we haven't really looked very carefully at that.
So it would probably have minimal side effects in the acute setting, because I guess, it could perhaps be limiting the immune response to other threats, which you're probably less likely to see in the 5-day window you're taking it for after an ICH, but I guess that might be more relevant for chronic administration at home in an elderly patient, who might get, HSV activation or something else like that.
Right, or a person who has BCID. That, in my mind, that's more of a chronic small vessel disease effect that would be more like Alzheimer's, where there'd be a little lower, it wouldn't be a big acute cytokine surge, it'd be a lower level of higher cytokines, but not as high as the big surge after injury. But yeah, in the 88 patients so far, we haven't seen any drug-related mortalities or drug-related SAEs, that are definitively drug-related. It's mostly
Yeah.
the disease progression itself.
Yeah.
Something happened.
It's fantastic. Halfway there. Well,
We're getting there.
yeah, nice. Right.
Timing of administration, particularly on the surgical intervention side, is something we talk about quite a bit. And Chris has been a global thought leader in pushing for earlier and earlier intervention, which might coincide nicely with the administration of drugs. How important is it for early administration of your medication? And it sounds like based on the safety profile, talking about sending troops to battle, maybe prophylactic administration before the injury takes place is something to even be considered.
That was my dream, was that our drug would become like the next low dose aspirin, where you take it every day and then you prevent the inflammation related, sequelae of aging or dementia. Who knows, maybe so, but boy, you can't get funding to do those kinds of trials. They're just super expensive. And even the A beta antibodies now, they were only approved so far for people who already have disease. But yeah, I think the way that it works, it would keep the cytokine levels at the physiological level where they're supposed to be. And so you wouldn't affect the beneficial responses of, the beneficial functions. And that's fine.
Okay. And this may be beyond the scope of this discussion, but as surgeons, I'm not sure we fully understand the potential benefit of evacuating an ICH. So I'm going to ask you to explain to me on a molecular level, what's the benefit of evacuating an ICH?
Well, because then you don't have that hematoma sitting around causing inflammatory responses to this. I mean, blood products they cause inflammation.
Okay. And is it synergistic with the mass effect of the hematoma, do you think, or is it primarily the blood products causing the secondary inflammation?
I'll be able to answer that when we do the pharmacodynamic measurements of the inflammation, because we have day 1, day 3, and day 5 blood samples. I'm going to measure them all at the end, so there's no batch effects, but we should be able to tell and then correlate that with the volume of the hematoma. So we should know, like, if they had a larger hematoma, did they have a much bigger inflammation surge or not? And we don't know if our drug will be more beneficial if they have a larger response, or if it'll be the same no matter how big the pro-inflammatory surge goes. I can vision that it could be either way.
As we were prepping for you coming here, I'm thinking about some of the other, just how useful something like this would be really across all of the ways we interact with neurologic disease, especially surgeons. And so the question, Brian, is, do you think this could be a surgical adjunct, maybe after minimally invasive ICH evacuation, or maybe just after, any brain interruption? I guess it depends on the processes that lead to inflammation after brain surgery, but we do give steroids for a week after most supratentorial surgeries, steroids for 2 weeks after most posterior fossa surgeries. You can imagine the havoc that wreaks on the patient's blood sugar and lots of other issues.
And that's with obvious evidence of harm with steroids, as opposed
And
to.
steroids, it's like you're taking a bulldozer. I mean, it's just so non-selective.
Yeah, I mean, the reason I asked you pointedly about timing is because it's hard to convince surgeons to operate acutely, even emergently on ICH, among other different diseases, and tying it to the administration of a drug that is also time dependent, I think would further provide an advocate for this type of ultra early intervention.
Yeah, I mean, if you have to think about, you have to look at the literature about when the cytokines go up and down after ICH. So, by about 24 hours or so, and then they tail off. By 5 days, it's basically back almost down to normal again. And so that's why we use that administration. But I can imagine you could even start probably 48 hours after, because the cytokines are still up by that time. But you wouldn't want to start 5 days after. So you'd want to at least have an acute intervention, but you could continue it. You could continue afterwards to look at some secondary sequelae, as long as you had a selective agent. You don't want to, my opinion, you don't want to continue steroids for that long, because then you're running the risk of influencing the the beneficial reparative processes that are going to go on.
And practically, you're administering the medication in your trial intravenously,
Yes,
but the bioavailability orally is also
yes. And
excellent.
in fact, one of the drugs in the radiation trial is oral. And our Alzheimer's drug is going to, the same drug, but it's going to be administered orally.
Like, it just, the practicality is limitless. If you think about maybe having a trial where you administer the medication before you intervene, because every neurosurgical intervention is a trauma of sorts to the
Yeah.
brain, and so limiting that secondary injury of even surgery would be
Yeah.
potentially beneficial.
So looks like the measurements that you're getting of the cytokines for all of these studies are peripheral blood, right? But I just
Or
wanted
CSF.
or CSF. I see. So do you find that they correlate pretty well, like is it, or not necessarily?
For some analytes they do, for some they don't. We just had a paper with Alamar recently on subarachnoid hemorrhage patients. This was a small pilot study I did with our CTSA, just to show that we could get samples, so match CSF and plasma over the first 3 days. And then we sent it to them to do their analyses. And sometimes the CSF and the plasma matched fine, sometimes not.
Yeah.
And in many cases, the plasma actually looked more consistent among patients than the CSF did. So it's hard to make a general statement about it.
Yeah. So it's interesting because I think, Lawrence Insting's work shows that there's some inflammation that's like in the brain specifically when you compare to peripheral blood. So, obviously eventually when you show clinical efficacy, that will answer the question, but I don't know if necessarily the peripheral blood markers correlate and showing everything that's going on. But the CSF it's
Right,
very interesting to
right.
have that.
And actually Lauren's on our
Yeah.
trial and working with her.
Yeah, you've got the CSF, and then you have the perihematomal edema to show, a
Yes.
direct radiographic
Right.
evidence of effect. There are two other disease processes I can think of, or maybe uses for something like this, and ischemic stroke is one of them. We're battling post-stroke edema on a regular basis. We just had a nice conference 2 nights ago about large core thrombectomy and the high rate of post-thrombectomy edema and hemicraniectomy after that. So it sounds like that would be potentially, an area that would be useful.
We didn't go forward with ischemic stroke from the beginning, mainly because we don't have any animal model data with ischemic stroke. I don't know why, but I just haven't found a collaborator that can do it. But I've got tons of compounds. So, if anybody has their own favorite animal model where they think inflammation is important.
I see a few students and residents in the ZIIC getting excited about a stroke animal model back there.
The animal model data there looks really good as well. That again is with Andy Blaskowitz. So he's got both ICH and SAH models. The SAH study is being funded by SBIR, just for preclinical work. And so now they've written a second SBIR to do a fast track to go into a trial, get ready for a trial. So yeah, it looked really good, and it actually blocked that secondary dip, that delayed cerebral ischemia type of dip. It blocked that. So
That's really interesting. And the other thing I can think of that's becoming more and more relevant is, whenever you place a brain implant in, you cause inflammation around that brain implant, and in many cases, it's led to degradation of signal for BCI. That was one of the main problems with BrainGate's device, the Utah array. And so maybe there would be an application there to minimize immune response in the presence of kind of foreign devices.
yeah. And since it is, it's stable and it's soluble, it would be easy to administer. You don't have to worry about organic solvents messing up whatever, wherever you're putting it.
Yes. I mean, could you foresee it safe enough to be administered as chronic suppressive therapy of sorts, specific to the
It's not-
brain?
It would.
Chronic immunomodulatory
Yeah.
therapy.
Yeah, keep it at basal levels, physiological. Now, if you have to have an upregulation of some reparative cytokine or some reparative, chemokine or something, maybe that, I don't know if that would block that or not. Because some of these cytokines have dual actions and, you block, let's say you block TNF alpha acutely, do you need TNF alpha later for some reparative thing? And if drug's still on board, would it block that? Or are the basal levels of TNF out there big enough? We haven't looked at any of those kinds of questions, but.
You commented on the glacial pace of the FDA regulatory process for drug development. And Chris and I only know device development, which if you have a predicate device that is similar to a new device, sometimes you can get it through in months without any human testing. So I would love you to comment on your perspective on the FDA regulatory process. Is it appropriate? And if you could change one aspect of it, what would you modulate?
Well, different divisions of the FDA are very different. So the neuro division is much more stringent than oncology, for example. I mean, oncology can get FDA, get a IND approval for things that would be so toxic that you would never be able to get the neuro group to say that. And yet many of these neuro diseases are just as devastating and just as important as cancer. But the cancer field has had a big start, and there was a willingness of the FDA to say, well, this is such a big problem that if a few regular cells get killed, who cares? Some of the early drugs like that for oncology were, very toxic. Some of the early drugs for Alzheimer's disease aren't very good either, but the FDA finally now has approved 2 of them. But we're a lot further behind the cancer field. And I think part of that, if there were changes to the FDA where they were a little more willing to take chances on some agents that might have liabilities, but at least they would be a start. Because if you can get something approved, then it's much easier to get the next.
To elaborate on that, you mentioned your involvement in the company that was spun out to further develop the drug and manufacture the drug. And we're always thinking about and wrestling with developing a vehicle to further bring something to market, but also remain on the science side to continue doing the work necessary to push it there. So how have you walked that line? It seems like you've done it well over the years.
The SBIR mechanism is really good for small companies because it does, there's SBIRs and STTRs, and it allows you to have academic partners. So you can marry the science and the business. And they're pretty good funding levels because it's a kind of a set aside. It doesn't go into the regular pool of grant reviews because they have a certain amount of money they have to spend on those grant mechanisms, and they don't always get very good applications. So that's a good way to get going and get feasibility. And if you partner with an academic institution who has experience or expertise in whatever it is you're trying to do, it's a nice marriage. And there's a certain amount that goes to the company and a certain amount that goes to the academic institution. And so that's one way to start and to get going on it. Of course, you guys have a nice advantage here because you have a lot of industry partners. That's really a benefit. It's important because SBIRs and actually grants, NIH grants itself. It comes a point where you can't get enough money to do what you need to do, and it has to be an industry partner.
Yeah, it seems like after this phase 2A, you'll have to get to a phase 3, and that
Yeah.
would be too big for
Right, I
NIH.
mean, even a phase 2B is probably gonna be too big for NIH, we're gonna have to partner, but what I'm hoping is that if our phase 2A study results look really promising, we're not powered for efficacy, but if there are trends in some of these endpoints, then I'm hoping that we can get industry interest. And even if it's not just for ICH, but the industry sees the potential for other diseases. I mean, there could be a huge market for lots of CNS disorders have inflammation as a component. And I'm hoping we could convince them of that.
Well, congratulations on all your work. Thank you so much for traveling here.
And thank
What an
you all for
exceptional talk.
coming.
Thanks, everybody.